So now we will come to a session that also considers pharmacokinetics, right? I mean, you have a mode of action that describes compound properties on targets, for example, or chemical reactivity and so on. But of course, the question is also how much of the compound do you have, right? So that's pharmacokinetics, and that's what Filip and other speakers in this session will talk about. Filip.
Yeah, thanks, Andreas, for the invitation and for organizing this event. On behalf of the team, I'll give an overview of how we have dealt with modeling human in vivo PK data and what were the challenges and opportunities we came across during that time. So when we think about in silico modeling, the majority of the tasks that we see in publications is the modeling of the bioactivity. So usually we want to design compounds that are active against certain targets, and this is one of the main focuses of machine learning. To a lesser extent, we are viewing or exploring off-target pharmacology and anti-targets such as hERG, CYPs, and to a much lesser extent, we are exploring the modelability of the drug exposure. And this component in drug discovery is equally important to the bioactivity prediction, but much less focus is put on predicting drug exposure. So our mission was to actually build machine learning models to see if we could guide early discovery using only chemical structure and dose. And in this sense, we aim to develop the human in vivo PK models. This would essentially drive the prioritization compounds for the in vivo experiments. So we will try to cut down the number of animal experiments that will go into to inform us about what to progress further or not. Also to design compounds with better safety profile and PK properties, and overall to improve the speed and efficiency of the design, make, test, analyze cycle. So this is the content of the presentation. I will briefly give a background around PK and also discuss the data curation protocol that we have developed. Then we'll focus on the modeling and the results, and at the very end, we'll discuss future applications. So AstraZeneca has several years ago defined the 5R framework, and this 5R framework is developed as a guidance on how to develop better drugs. So this consists of the right target, the right tissue, the right safety, right patient, and right commercial. And the PK component actually incorporates at least two of these parts of the framework, the right tissue and the right safety. And I would like to focus on several components of the pharmacokinetics that are essential for the drug exposure. One of these is the absorption. As you know, majority of the developed drugs are intended for the oral administration, and each drug project developing such drugs aims to have high oral absorption and bioavailability to achieve optimal exposure in vivo. Another important component is how this drug, once administered, how does it distribute into the tissues and organs? So does it reach the intended target? And this is what we measure with the volume of distribution. And at the very end, we would like to see that the compound gets eliminated properly. And usually we measure metabolic clearance. And we aim for low metabolic clearance to have acceptable duration of the compound within organ. At the very end, bridging between the efficacy and the safety is Cmax, which basically should be in the therapeutic range to achieve the needed efficacy, but also stay beyond the toxicology or the upper safety level. When it comes to the data that we've used Pharmacokinetics PK module. It basically collates all the FDA and EMA documents that have become available with decades of medicinal chemistry research. And we started with around 1,400 drugs, and we employed a fairly rigid and a fairly, I would say, fairly different point of the curation protocol to deliver the best data possible for the modeling purposes. So together with the experts in the field, we have put a particular focus on including adult healthy volunteers, but also certain patient groups where we have compounds developed for these indications. These are primarily the oncological patients. We focused our studies on the single dose PK for small molecules and also on non-tissue specific measurements. What we have excluded were the inadequate patient groups. So these were the patient groups that had signs of decreased PK functionality, such as liver cirrhosis, renal dysfunction, etc. As machine learning essentially is not able to deal with the incomplete data, we had to remove the incomplete data points and also remove the measurements that were related to the radio-labeled compounds, metabolites, and enantiomers, but also incompatible assay technologies and concomitants that may affect PK. At the very end, we have obtained data for 9 different PK parameters, and as you can see to the right, these can be divided into 12 PO or oral and IV intravenous variants. Some of these parameters have been measured for both PO and IV, and these are AUC or area under the curve, half-life, and renal clearance, while others were exclusively measured for IV or PO, as you can see in the Venn diagram. As for the data aggregation, we use the median value, and we pay particular attention to keep compound dose combinations as our point of reference for the modeling, and the doses, because we use doses as well. The doses were rounded to 2 decimals if below 10 milligrams or 0 if above 10 milligrams. At the very end, we obtained around 1,000 unique compounds for these 12 PK variants, which can be then turned into around 4,500 compound dose combinations. If you look at the data matrix that we obtained alongside these 4,500 compounds and 12 PK parameters, we can see to the right that we have a varying level of the data completeness. Some of the parameters, such as Cmax per oral, had a maximum number of data points, 67% still of entire matrix, while others, such as the renal clearance IV, had 159 data points. And here we can observe trends that are fairly obvious in drug discovery. So, for example, the PO parameters were more frequent than the IV parameters, and this is largely due to having drugs developed for the oral administration purposes. Some of the data, such as the bioavailability and the clearance, were biased towards the optimized values. So we frequently saw that we had many compounds with high bioavailability, which is expected when the compounds come to the clinical stage, but also low clearance, so the drug could achieve desired exposure. And overall, we can see that for some of the data points, we had limited data for the model building. As far as it comes to the feature designs that we've used to build the machine learning models, we have explored several. We've used only dose as a single feature, and this was to investigate the predictiveness compared to other more complex features. On the other side, we also built models with ADME and PK in silico predictions that come from our internal models. This also incorporated the model of the in vivo rat PK, very classical proxy structural finger descriptors, such as the 2D descriptors and 1D descriptors that we call OS-Selma were used as well. And at the very end, we combined all three to see how a concatenated version would behave in terms of performance compared to the others. We termed it combo FP. We tried several machine learning methodologies. They more or less gave same or equivalent performance. We chose random forest as the modeling method, and this was what we reported in the paper. We used fivefold cross-validation on 80% of the data that we designated as a training data, and this data was also used for the hyperparameter optimization. And then the finally derived models with the optimal set of hyperparameters were used to assess 20% of the data that were taken as a holdout test set. We have also developed the custom data split for validation and the test sets. So we made sure that the compound with all of its doses was part of the same fold or part of the same set, sort of not to influence or to leak the data, and to actually have the models that learn different chemical structures, so the chemical structure would not be shared between the folds. To the bottom, you can see the in vitro ADME and physchem properties that we have used, as well as the in vivo rat PK parameters, and these are very typical for the early stages of the drug discovery. So they overall assess the lipophilicity, the solubility of the compounds, permeability of the compounds, and also metabolic clearance in vivo. So all these features, either separate or in combination, were used to build the models and to predict 12 PK parameters. So having said that, the cross-validation was designated the same way as the test set, because we have been splitting the data not to leak the compound information between. We first evaluated the cross-validation results, and you can see for each of the bar plots we have the error bars as well. For a good portion of the parameters, if we look at the R-squared and we put a threshold somewhere at 0.4, we can see that we observe quite fair performance for 7 or 8 of these parameters. And this was mainly achieved using combo FP, but in certain cases, ADME PK properties itself were sufficient to drive the modeling. In terms of the RMSE, it's a bit cryptic because we are at this stage not aware of, we have not included the experimental error of measurement to assess, but for some of the parameters, we can see that we have quite low RMSE, like Tmax PO, for example, and for others, slightly higher than expected, like for clearance. Of course, we needed to take a look into the performance that is done on the holdout test set, which was more or less following the same trend as for the cross-validation. And here I only show the RMSE performance, but I add 2 more bar plots. These bar plots actually present a mean predictor. This is a dummy model that takes the distribution of the training data and takes the mean value out of it and then assigns the mean value to every test instance, and then we can evaluate the model. So this was, we envisioned this as the point of the lowest performance we can go with the models, and it would be basically what if the model only learned the mean of the distribution. On the other hand, we have the opportunity to estimate the experimental error of the measurement, and this was done as a 2 times the median value of the standard deviation, and this was the point of our highest achievable performance. Unfortunately, if we would compare the performance given in dark blue, and we simply estimate whether we were closer to the mean predictor or to the experimental error, we would see that only for 3 PK parameters, these are the AUC-PO, Cmax-PO, and VDIV, we will see a performance that was much closer to the experimental error. So these were the only 3 models that we progressed further, and we found these to be reasonable for the use. Of course, to further estimate the model performance, we need to look at the correlation of the parameters and the predictions. And as you can see, for all 3 PK parameters that are found here, and their holdout test sets, we see a quite wide range of the experimental values, which is typical for all stages of the drug discovery. And equally, the predicted values were spread out quite evenly, quite well. Best performance was achieved for AUC-PO and Cmax-PO, as you can see, based on the correlation plots, and to a lesser extent, the volume of distribution ID. However, all 3 models are fit for purpose and to be progressed for further analysis. Of course, the random forest is convenient for several reasons, one of them being able to extract the feature importances. And as you can see, for AUC-PO and Cmax-PO, as dose-dependent PK parameters, the dose was the main feature responsible for driving the model predictions. But however, other ADME and PK properties, such as predicted Cmax from rat, were also important, which was in line with what we expected, having animal species involved, but also in vitro experiments. On the other hand, volume of distribution had predicted rat volume of distribution as the main feature, and then other components describing 1D and 2D descriptors of the chemical structure were found to be most important. We had external validation, and this was our internal AstraZeneca compounds. So that was a few years ago, there was a paper discussing the clinical development of the AstraZeneca compounds, where 126 compounds have become available from the clinical data with the dose and predicted preclinical PK parameters using DMPK methodology. And here we have compared for the parameters that we were interested in. We compared how the machine learning models compared to DMPK derived estimations. And as we can see from the RMC values, in all cases, DMPK had estimated overall better, had better prediction power than the machine learning models, except in one case, and this was for the volume of distribution. But please note that here are only 5 compounds where we have volume of distribution available. So this is not statistically valid, but still, the DMPK measurements are cumbersome to obtain and to get the estimate. And at that point, we already have compounds synthesized and progressed. So we can fairly say that the machine learning models are, based on these results, fit for purpose to be used in early drug discovery. And at best, they are also complementary to the current preclinical predictions. So both of these modeling techniques can be used together. So for example, use machine learning models to pick the right compounds with the right profile, and then later that would be reconciled by the DMPK observations. As for the potential model applications, as said, the intention of the models are to be used in early drug discovery, and the primary component is to increase the efficiency of the DMTA cycle. So with these models and others that we have in our collection, we influence the decision-making of what compounds to send for the synthesis and also for the experiments, and that would cut down unnecessary number of iterations, and we would quicker come to the clinical candidates. But on the other hand, because we are dealing with AUC and Cmax, these are also important for estimating safety-related models and visualizations. So, for example, we can utilize to predict predicted Cmax to estimate the exposure in organ toxicity models, etc. For the conclusion, we have established a very comprehensive protocol for the curation of human in vivo PK parameters that were then investigated using machine learning models. Through rigorous evaluation, we were able to obtain 3 fit-for-purpose models that were successfully validated using internal clinical data. And although the DMPK modeling showed better performance, their development is still cumbersome and requires lots of data, which still plays into the strengths of the in silico alternatives. The paper was published about 1 year ago in Molecular Pharmaceutics. I invite everyone to read about it. And the model that we have also used to drive these models, the RAT-PK model, was also published in Molecular Pharmaceutics around the same time. Our models, as said, we believe that they have great potential to assess the candidate prioritization, but also aid individual project decisions, and in some sense also guide the novel generative models as through the multi-parameter optimization protocols. At the very end, I would like to thank all the people that have been involved in this project, and I'm happy to take any questions you have.
Excellent, Philip. Thanks a lot. Are there any questions from the audience? The paper is linked from the event website, by the way, in case you want to read up in more detail. So let's see. Yeah. What I'm actually very happy to see is that there's always the question, if you build models, right, how physiologically relevant is that actually? Yeah? I mean, do you predict proxy endpoints or do you predict something in vivo relevant? Well, and here the answer is quite clear, right? Because you model the in vivo endpoint directly. Yeah. So that is, I'm glad to see that, because then you don't have those models of model situation, right? As one always calls it, right? But you have a model for relevant endpoint directly.
Yeah, it's true.
Okay. Are there any questions from anyone? Okay. Yeah, there don't seem to be any.
If there are any questions, they can reach. Anyone can reach me out on LinkedIn. Please feel free to do so.
Sorry, there is a question in the chat.
Yeah,
that's right. I didn't see that. That's right, Boris. Yours is the first one, right? And Mohammed has one as well. Boris,
yeah.
Go
ahead. Should I read it aloud?
Yeah, because then you can elaborate a bit.
Live,
okay. The question from my side is, what do you think? What's the reason of the machine learning performance? Why it shows the worst quality in comparison to the PMK, if I spell it correctly? Yeah. Like, is it due to the data, available data, amount of data? Like maybe it wasn't enough, or maybe it's due to statistical distribution, data properties, or there is another way, is because of the method, because of the algorithm you've chosen. Like, I mean, random forest has some drawbacks, and maybe that was the reason. Yeah, thanks.
So thanks for the question. When it comes to the random forest, it overall showed quite fair performance compared to all the other methods, and we have seen no clear benefit of using more sophisticated methodologies. When it comes to DMPK, at the time when DMPK estimates are done, this is when all the in vitro and in part in vivo experiments are derived, and usually these are estimated from triplicates or even more data points available there. Another component that is important there is that DMPK measurements, they do not have always estimated equations that are used, but there's at certain times more customized to fit particular purpose. So this could be also the reason why DMPK measurements are, and they also come from years of knowledge-based estimates, estimations, and work. The models here also do not tackle, or they don't take in consideration different compartments. And this is what the DMPK models and DMPK equations also take in consideration. So different compartments and how the drugs are distributed are also essential to drive more precise estimates using DMPK.
Okay. In the end, it means that if we fit this information or something correlated with this information you said into machine learning model like random forest, then we can get better results for ML model. Yeah.
Well, that is theoretically something that can be worked on. That would be theoretically possible as well. So it's one of the main discussion points, also how to incorporate the known DMPK approaches into machine learning and how to simplify the work that DMPK folks are doing during the drug discovery.
Okay, thanks.
Thanks.
Great, thanks, Boris. And I think there are two more questions actually, but in the interest of time, I think Philip, maybe you can follow up in the chat to everyone, so everyone knows the answer. And anything you wanted to say about the journal as well, or?
Yes, about the journal. So I wanted to invite people to contribute to the journal. This is a recent journal. It's called Artificial Intelligence in the Life Sciences. It's focused on the modeling aspects of just about anything that you can find in drug discovery. It's open access journal. And this year, the fee is waived. So I would invite people to use the opportunity to consider it for publishing. And in particular, I'm a guest editor to early career researchers in AI, which I think it would offer amazing opportunities to young researchers to be in the driving seat. So as corresponding authors and driving their paperwork. So please feel free to, I'll leave the link in the description in the chat so you can visit it. Thanks.
Excellent. Absolutely. Please do that. Yeah. Great. Thanks a lot, Philip, for your contribution. And we will stay in the PK area right now. We'll move to Ross Kelly from Syngenta, who tells us how kinetics is used in regulatory assessments as well. Ross.
Thanks, Andreas. If you can hear me okay.
Yep.
Good. So, yeah, thank you. So, yes, I'm going to be talking about using PBK models to reduce kinetic uncertainty in regulatory risk assessment. So first of all, thank you for the opportunity to speak to you all today. Lovely to be here. I'd like to give you a flavour of how we use PBK models within Syngenta, particularly in late stage work, and then specifically what our current approach is with respect to reducing kinetic uncertainty, looking at risk assessment. So what I'll do is I'll start by giving some generic PBK considerations, how we select models and how we evaluate them. And then I'll go through an example project, which will include things like creating a chemical specific rat PBK model, how we extrapolate this to human, making some population variability simulations, and then hopefully contextualizing this internal exposure model outputs with respect to kinetic uncertainty. So for those of you that don't know what PBK is, PBK stands for Physiologically-Based Kinetic Models. So we drop the Pharmacokinetic bit in agrochemical. So essentially these are compartmental models used to simulate the time course of a chemical ingested throughout the body. And pictures are always an easy way of looking at these things. So on the right, we have a schematic of what a PBK model can look like. So as you can see, they are anatomical in their representation, and they are comprised usually of organ-specific compartments. So we have lung and gut, etc. What we can do is we can lump some compartments together. So we have a rest of the body compartment here as well. And these are all linked by blood flow, akin like it is in the body. So PBK modeling comes into its own with respect to physiological detail. So we can capture absorption, distribution, metabolism, and elimination processes. So they're just a little more involved than compartmental PK models. And aside from general modeling advantages of developing and testing new hypotheses, etc., there are some specific advantages to taking a PBK approach. So first and foremost, they allow us to make tissue-specific predictions, which is very useful. They also allow us to extrapolate between species, which is useful given that we can't test agrochemicals directly on humans. And they also provide a basis for reduction of animal testing, which has been mentioned a few times today as well. So they can also help us when we have different types of data. So if we have minimal data, we can use them to rank compounds. If we have chemical specific data and ADME data, we can use them to rank compounds. So just wants to move on to the next slide. There we go. So thinking about PBK models in terms of risk assessment, there's a generic description of what risk assessment is at the top, but typically risk assessment aims to evaluate the magnitude and the frequency of human exposure and estimate the potential adverse consequences. So we can have some uncertainty and variability within these data, so interspecies and intraspecies variability. And these are usually accounted for using uncertainty factors, and these cover things like insufficient data, dose and duration, different routes of exposure, extrapolation across species, and population variability. And kinetic processes can vary between species, they can vary between dose routes and dose levels, and so it's important to understand and consider how the dose response of a chemical can differ across populations as a function of kinetic processes. So default uncertainty values decrease the amount of chemical that we can apply. So there's an uncertainty factor of 100-fold here. And we hope that we can use PBK modeling to help us address this. So the use of PBK models, fortunately for us in regulatory risk assessment, is becoming more prevalent and it's actively encouraged. So the OECD published a guidance document that provides a framework to bridge between PBK modelers and regulatory assessors. And so the ultimate motivation here is to reduce these generic uncertainty factors when possible. So that will allow us to apply the maximum amount of chemical safely at the coalface. So now we all know what PBK models are and what they can do. We can have a look at and think about how this works in terms of risk assessment. So we can look at the uncertainty and extrapolation. So in contrast to classical kinetics, PBK modeling provides a basis of extrapolation that's scientifically grounded. So, for example, we might have some kinetic data available for healthy rats, but the risk assessment may wish to consider children via an inhalation route, for example. So obviously we can't test the compound on humans, let alone children, and so we have to have an approach that can help us tackle this. So a PBK model can explicitly incorporate age and route-related differences in ADME, as well as other things like biochemistry and physiology. So the next thing we can think about is model structure. So when we evaluate model structure, we're looking to assess confidence in model and like the problem formulation. And we must carefully consider a model, and in this instance, we'd say that being published isn't necessarily a good enough justification for model selection. We need to evaluate the biological suitability of a model. There's loads of these types of models out there, and we can create bespoke ones as well. And needless to say, as usual, the model structure needs to be biologically relevant while not overly complex as well. And then finally, this is a bane of most modelers' life. We need to determine the verifiability of the mathematical description. So if we can seek original model code and simulation scripts, that's preferable. And as we know, recreating a model from a journal paper is nearly always problematic when we're dealing with units and missing parameters and clarity around scaling and metabolism. So that's something that we consider. We also consider evaluating the model parameters. So are the parameter values consistent with well-vetted collections of parameter values? So there's forms for tissue weights, cardiac outputs, and these tend to be well reported. Are the parameter values uniquely identifiable? Have variability and uncertainty been characterized? And this is something we can look at ourselves as well. And then finally, we want to look at model performance. So the ability to predict the observable data when we have it. So we can have a qualitative look at this, trends with respect to changes across conditions. So the direction of the deviation from the model from the data. So we're having an under prediction or an over prediction. And then we can also look at quantitative trends. So the magnitude of the deviation between the model and experimental values. So the IPCS, the International Programme for Chemical Safety, in 2010 suggested that in PBK modeling, predictions that are on average a factor of 2 away from the experimental data are considered as adequate. So I guess that's for the jury to decide whether we accept that or not. And then just some minor considerations as well with respect to risk assessment. They're not usually interested in black box models. They want to be able to have a look under the bonnet and see what's going on. So software availability is also an important factor for selection. So when we're taking a PBK approach, we're looking to predict an internal exposure metric. So rather than comparing external points of departure, so at the top left, we've got a rat oral point of departure. We're looking at an internal metric. So we're trying to understand the kinetics so that we can look at these the default uncertainty factors. So without modeling, we have some approaches that we can take. So they can have a margin of exposure, just looking at the rat oral point of departure and a specific consumer exposure. And with modeling, we can go one step in, we can go through different species, and we can try and get an internal metric. So we'll come back to this slide as this forms the basis of what we're really trying to achieve, but we're just looking for a better understanding of the kinetics to address that uncertainty. So I'll take you through an example of where we've used PBK modeling to predict these internal exposures in an attempt to address these uncertainty factors. So we have a particular chemical where we have a series of acute and dietary operator exposures for population subgroups. We also have some in vitro data for rat and human, and we have some in vivo rat data as well. And then we're going to combine all this and try and do something useful with it. So for this chemical, we opted to use a version of the Sheila-Peters model. So this is a published model, which is a 14-compartment PBK model. It's a little more involved than the ones I've been displaying in the previous slides. And the somatic component of this model includes multiple compartments for the blood and different tissues in the body, as you'd expect. So this is also expanded to include an ACAT component, which is an advanced compartmental absorption and transit model. And this is to more accurately model or describe the fate of an orally ingested chemical. So this component allows us to capture the process of dissolution and absorption in the GI tract, which is useful. And then just thinking about describing model selection, is it biologically relevant? Well, we think it is with respect to what we're interested in. Are the parameter sets in good accordance with the literature? So there are several parameter sets that are in good accordance with this model, and it also usefully has parameter sets for a rat version and a human version, so that's good. The code is also openly available. You can get a copy of this model through Symbiology, I believe, and it's also highly reproducible. So we start with a generic PBK model using the generic parameters. And we input the chemical specific in vitro parameters and the ADME parameters, and we see how the model fares up against the in vivo data, the rat in vivo data. Now, realistically, it's not a bad start. We're in the right sort of ballpark, and if you're thinking about that twofold factor, then it's probably not that bad at all. As I said, the models are originally based on this Peters model, and there was a useful accompanying paper that described a series of approaches that you can take to align the line shapes of your model output to the data if they don't align. So the framework suggests, looking at solubility and the absorption of the profile, that the in vivo rate of permeability is greater than that used in the simulation. So it just says we really need to look at using a higher permeability rate. So the next step is to use that in vivo data to generate a chemical specific version of this rat model. And that's what we did. So as always, we're looking to make as few changes as possible. And we conducted some just bog standard local sensitivity analysis on a small set of the physiological and chemical specific parameters to see the effect that it had on the venous simulation profile. And this was based on the internal data that we have for these. Some of them had a questionable error score, so it was a good thing to have a look at. So some parameters are more sensitive than others. The blood to plasma ratio is quite sensitive, but the values that the simulation include are super physiological, so that's irrelevant. And unsurprisingly, the effective permeability, this PF at the top, seemed quite promising in order to get that model to align with the data. There's two scaling factors here, which I won't talk about, but they also seem to be important. So following the sensitivity analysis, we estimated the chemical specific parameter set for this model, and it involved 3 relatively small changes of these top 3 parameters here, which was good. And the model's now well aligned with the data with these small changes. And we also noticed that changes in rat body weight also played quite a big role in the venous profile. So we estimated some parameter values to reflect the upper and lower bounds of the body weights based on the data. So we're interested in modeling human oral exposure, so we have to make a few jumps to get there. We've got rat in vivo gavage data, and we've modeled that. the next step is to adapt the model to reflect a dietary intake of the active ingredient. So we did this by utilising a study done by Wan et al., which was where they looked at rat food consumption quantified at 30-minute intervals over a 24-hour period, which I believe is this solid circles on here. So what that means is we can pick this up, we can split our dose into these increments and mimic a dietary intake, and this will allow us to simulate dietary consumption of the chemical based on that single dose. So the doses we look at going forward are the no adverse effect levels based on a 2-year rat study. So we have a dose for male and female. And then what we decided to do was introduce some variability within the model. So we're looking at intraspecies variation. So again, we're trying to understand the kinetics and trying to make some prediction on kinetics, looking at that uncertainty down the line. So specifically, we introduced a 30% deviation from a series of parameters, including body weights, flow rates, clearance, and absorption. And then simulations gave us a profile for the variability with respect to the dietary consumption. So a red line here indicates the median. We have grey lines, which are the individual simulations, and then we've got an upper and lower percentile bound. So this gives us a population view of the internal exposures for the rat. And then, as I mentioned initially, we need to be thinking about the reliability of this kind of models. Now we're making some jumps, so we've got to be careful with this. So we then looked at the uncertainty and sensitivity of the parameters. Now, I'm not going to go into a huge amount of detail with respect to the methodology for this, but for brevity, we calculated a sensitivity coefficient, which is this SC on the left-hand side. And this essentially represents a fractional change either in AUC, which is what we've used here, or Cmax, as a function of a fractional change in a parameter. And so we can rank these as low, medium, high in terms of sensitivity. And then using the previous intra-species variability simulations, we can calculate an uncertainty metric, which uses the median and the 95th percentile results following some Monte Carlo simulations. So this gives us our uncertainty sensitivity matrix. And the key here, as always, is that if we have highly sensitive parameters that are also uncertain, then the reliability of the model is questionable, particularly with risk assessment. So gladly we didn't have either of those. So the next part of the process is to find out what the human equivalent dose is. So we use reverse dosimetry to predict the dose required to yield the same equivalent AUC from the dietary rat study. And we opted for a human feeding schedule looking at this graph at 8 AM, 1 PM and 6 PM. Now, I certainly don't eat at 3 specific intervals every day, and an interesting result was actually the model can be quite sensitive, or the outputs can be quite sensitive to variations in this. So that's something to think about in the long term. But in terms of jumping from the rat model to the human model, as I mentioned, the Peters model has parameter sets for rat and human, but the estimated scaling factors for the partition coefficient and the renal elimination, we conserve those. So there's some alignment between the rat and the human model. And then again, we looked at the uncertainty and sensitivity matrix to give us some sort of thoughts on the reliability of the model. So modeling human population variability is slightly more complex than rat. So we used a package or a function from the high throughput Toxicology package, the HTTK package, which samples from the NHANES database to generate age-specific parameters. So the NHANES is the National Health and Nutrition Examination Survey, and it involves an interview and a physical examination where they take metrics like this. And there are some regression equations published in the literature which can go from things like sex, age, weight, and height, and take us through to estimations for tissue masses, blood flow rates, and things like that. So what it allows us to do is generate a range of population parameters, and this allows us to create a virtual cohort, in essence, of people to simulate, which is what we're interested in doing for those population subgroups. And then there are also other things that we change. So you can look at intrinsic clearance, and there was a paper that suggested this can decrease by 0.8% per year after the age of 40. So it's a bit more involved, but this is in essence what we did. So why are we interested in this? So recall, we're interested in understanding the internal exposure of a series of population subgroups. So with our population samples, we were able to simulate a time course profile for acute and chronic for each of these. And we've now got a predicted internal exposure. And again, we've got a median and an upper and lower bound, so we've got some variance in the model outputs. So this is for chronic and acute. And we can take these forward and we can bring this back to our schematic of internal exposure and use this information to try and do some analysis. So we're interested in the margin of internal exposure first and foremost, which is a metric that is simulated by the rat dietary no adverse effect level AUC divided by the population simulated mean AUC for each group. And what this boils down to is it suggested that if you need to have a value of 25 as a minimum. And so on the log scale, you can see here that we far exceed this. So what does this mean in reality? Well, these results tell us some different things. So if we take, for example, the children population aged 1 to 2 for chronic, and we have a look at the thresholds, so the margin of internal exposure far exceeds that 25 minimum threshold. However, the margin of safety, which is another metric, doesn't. So it implies that by understanding some of the underlying kinetics in the model, we can help give confidence to different kinetic safety metrics. So the final part of this model was to look at the impacts of the PBK outputs on these kinetic uncertainty factors. So these uncertainty factors essentially account for poorly quantified influences. So we can have things like interspecies differences, i.e. the uncertainty moving from a rat to a human, and inter-individual variability, so uncertainty within a population. So we're only interested in the kinetic differences here, although we have done some work for the dynamics. And what we can do is we can now use the inputs from our simulations to calculate some of these data-derived extrapolation factors that we can inform on. So we can take an animal point of departure, which we have, and the human equivalent dose, which we've simulated, and we can calculate this. And then we can look at the inter-individual uncertainty factor, which is below. And again, these outputs come from our model simulations. So if I remember rightly, the AUC the general population, we assume to be a healthy population of 20 to 49-year-olds from the US population in the simulations. So if you follow these through, you can calculate a composite factor. You can assume that the dynamic factor is still 10, and then we can calculate a reference dose. And we're interested here in the percentage of the reference dose occupied by the acute and chronic exposures. So these I haven't showed all of them, obviously, because there's no need, but the percentage of the reference doses are incredibly small, or the model predicts that they are. So this tells us that some of the internal exposure is extremely small for some of these operator exposures, and in some cases it's less than 0.1%. So by taking this approach, what we're really doing is trying to understand and model the kinetic risk, which we think here is to be small. So just to conclude, we looked at some general considerations for PBK modelling with respect to risk assessment. We've shown an example where we've taken a whistle-stop tour at that example of taking a rat PBK model that's generic, making it chemical specific, extrapolating to human, doing some population variability, and then using those internal exposure metrics to prove or to suggest that we understand the kinetics a little better. And this is ultimately helping towards trying to address and reduce those default uncertainty factors. So that's everything today, just to make some acknowledges, and thank you very much for letting me give a talk.
Yeah, thank you, Ross. Much appreciated. It was a very nice overview as well of different approaches, applications, and so on. Excellent. Are there any questions from anyone or any comments? Nope. Okay, because if there's not, I cannot. Yep, yeah, Julia, yeah, please. Do you have a microphone? Otherwise, you can type it as well as you prefer.
Oh, yeah, hi. Yeah, sorry, the typing took a little bit longer, so I thought this might be better. Thanks so much for this talk, Ross. I recently saw a newly published paper by Brown et al., and they were talking about their opinion that PBK parameters may be inherently and practically unidentifiable. So they were talking about their observation that the parameters that you select might not be a unique set of parameters. So I was wondering, what's your take on that? What's your experience on that?
Yeah, thank you for the question. Nice to speak to you, Julia, again. Yeah, so I think that identifiability, I've only really experienced it with compartmental PK modeling, where you can get, big differences in the absorption rate and the elimination rate. Personally, I've not looked into it in too much detail for this. I know others in the group do, but it is certainly something to be aware of. And I mean, we're always trying to address uncertainty, and we're making some big jumps here in terms of moving from species and assuming different dosing regimens, etc. So I think we can just add it to a list of things that we're uncertain about, but it's definitely something that we'd look at. If you could send me a link or point me in the direction of that paper, that'd be great as well.
Yeah, will do. Thank you.
Thank you.
Cheers.
Yeah, cheers, Julia, for your question. And George Loizou says, great talk, Ross. Thank you. Much appreciated.
Thank you.
Okay, great. Thanks a lot, Ross, for your contribution again. And so in the next talk, we will actually focus on a particular compound and using transcriptomics information as well as PK to come up actually with guidelines for the safety or risk of that compound. And our speaker will be Arthur, who will comment on that.
Hi, Andreas. Hello, everyone. Can you hear me?
Yeah, sounds good. Yeah. Feel free to share your screen and then we can see that as well.
Okay, thank you. Can you see now?
Yep, that's right. Yeah.
In presentation mode now?
Yep, that's right. All good.
Okay, thank you very much. And good afternoon, everyone. My name is Arthur. I'm a computational toxicology fellow based in the University of Birmingham and sponsored by the Food Standards Agency, and also working in a partnership with the HSE Science and Research Center. From now on, I'll present our recent work, which is an in silico workflow integrating physiologically-based pharmacokinetic modeling and in vitro transcriptomics data to derive a health-based guidance value for perfluorooctanoic acid. So perfluorooctanoic acid, and as we will refer to it here in this presentation as PFOA, is an economically significant chemical for companies producing fluoropolymers and products such as stain repellents, polishes, coatings, and paints. It is also a surfactant, and it presents a backbone of carbon-fluorine atoms. And it's worth to mention that this bond between carbon and fluorine is strong and stable, and this reinforces how persistent in the environment this chemical is. As many of you are aware of, it is also known as a forever chemical. Besides, PFOA is also a xenobiotic, a carcinogenic agent, and an endocrine disruptor. Exposure to perfluorinated chemicals in general is linked to a decreased antibody response, dyslipidemia, decreased infant and fetal growth, and increased risk of kidney cancer. And PFOA exposure came also to the spotlight due to the public case involving DuPont facilities and contamination of drinking water with PFOA in the United States. And it is very well presented in the movie Dark Waters for those interested in this movie. The main goal of this work was to use transcriptomics data obtained from human liver spheroids exposed to PFOA as input to our workflow to derive a health-based guidance value for this chemical. So initially, it's important to mention that tissue dosimetry can be estimated with the application of physiologically-based pharmacokinetic modeling, or PBPK, and it was very well presented by previous speakers, Ross and Filip. PBPK modeling is a powerful means of simulating the factors that determine tissue dose within any biological organism and consequently its correlation with health effects. The value of PBPK models is that they are tools for integrating in vitro, in silico, and in vivo mechanistic pharmacokinetic and toxicological information as well. And PBPK models can also encode an explicit mathematical description of the important anatomical, physiological, and biochemical determinants of chemical uptake, distribution, and elimination. PBPK modeling as well is increasingly being used in chemical risk assessment. Initially, in the modeling step, simple but realistic models can be generated, and we can increase the complexity of each model as soon as it is required. For example, here in the left-hand side in this picture, we can see in the bottom part of the picture, rapidly perfused and slowly perfused compartments. But initially, we can remove the liver from the rapidly perfused compartments because of its major metabolic capability, as well as the fat compartment. We can remove it from the slowly perfused compartments because the vast majority of chemicals in the environment are lipophilic, and they will eventually accumulate in the fat compartment. In the right-hand side of this slide, the graphic that is animated is demonstrating tissue dosimetry as one of the abilities of PBPK modeling. And it will allow us to analyze what is happening in different compartments after exposure to a given substance over time. So as we can see, these compartments represented here, brain, kidney, muscle, fat, and liver, they are dealing with a chemical in a different way. Some of them are washing out the chemical over time faster than others. And we can see that muscle and fat compartments are accumulating this given substance over time. So as an effective way to perform reverse dosimetry, we have two ways to approach this. The model can predict concentrations in the cell, which is the tissue dosimetry. And the in vitro concentrations, they will serve as a surrogate for the in vivo tissue concentrations. And therefore, we can extrapolate the concentrations observed in vitro to what individuals are exposed in the environment. In this example here, we have two arrows, one red and one green. The red arrow is representing what is the most familiar process. We're seeing here the forward dosimetry. And on the contrary, the green arrow is showing the less familiar process, which is reverse dosimetry, because we are starting from the tissue dosimetry and then trying to extrapolate to the exposure dose. And as soon as we have the tissue dosimetry information, we can analyze several things. Among them, we can analyze the altered gene expression. And because of these altered gene expression patterns, we can identify differentially expressed genes, link them to perturbed biochemical pathways, and hopefully link them to metabolite signatures that can give us the full information that we need to link everything and put everything together to better understand a toxicity or a disease outcome. So this is an overview of what we have done here in terms of each step that we conduct in this work. So in the step one, we obtained omics data related to the in vitro exposure of PFOA. After we tried to derive a molecular point of departure using benchmark dose modeling, and then identifying genes that had concentration response relationship, we tried to estimate the calculation, try to do the calculation of in vitro free concentrations, then use those in vitro free concentrations related to that gene that has those response relationship, and then used as an input to our PBPK model and to our algorithm for quantitative in vitro to in vivo extrapolation. And ultimately, we try to generate BMD lower confidence limit of molecular point of departure. And as a data source here, we used a study that was published by Health Canada, references down here. And in this study, they had exposed human liver spheroids composed by primary human hepatocytes and Kupffer cells. They have exposed them, these liver spheroids, to 4 perfluorinated chemicals. And they are PFBS, PFTS, PFOA, and PFOS. Our main focus was perfluorooctanoic acid here. We just focused on that. They used 10 concentrations from 0 to 100 micromolars. The top non-cytotoxic concentration to the liver spheroids was 20 micromolar. They used 4 time points, day 1, 4, 10, and 14 of the exposure. They used also targeted RNA-seq panel with 3,000 genes representing a diverse set of toxicologically responsive pathways. And as an important information, they state that the day 10 was the best time point to observe differentially expressed genes for PFOA. So we have focused here then on day 10, 20 micromolar. We got the 4 replicates for these related to this concentration and the 4 controls as well, just to have an initial analysis on this data. Afterwards, we used the list of genes of interest as an input to BMD Express tree, where we performed a benchmark dose modeling and a functional classification. After conducting the benchmark dose modeling, we selected the best BMDL model and then used the concentration response data related to this best BMDL model as an input to our quantitative in vitro to in vivo extrapolation algorithm. From the PBPK side, the PBPK model was already published by Dr. George Louisou and his group. And what we have done here was, first of all, choose a time interval along the region of the steady state of before exposure. And in this case was equivalent to 11 years of exposure. The initial study had a 35 years of modeling. They were modeling at 35 years of exposure, 30 years of exposure plus 5 years of washout of the compound. And here we were proving a concept, so we just selected the beginning of the steady state just for computational time processing sake. And we also tried to make sure that the demographic of the population chosen was able to reflect the population exposed to P4, reflecting very well the data analyzed during the biomonitoring studies conducted in the area of DuPont facilities back there in the States. And afterwards, it's important to mention as well that we had calculated the free in vitro concentrations, and we did that using the Armitage approach, but we could also use the virtual cell-based assay because they basically work the same. They will both compartmentalize the assay, like the microplates. They will account for the headspace of the well. They will account for the serum lipids proteins in the medium, the bulk medium, the cell line, and as well as the cellular uptake, the unbound chemical in the medium, the passive diffusion and the cellular excretion, the intracellular protein binding and lipid binding as well. So we used the nominal concentrations, the physical chemical information and data related to P4, and the details about the assay, the in vitro assay we were trying to mimic here to finally estimate the free concentrations for perfluorooctanoic acid as well. So after the initial filtering, we have filtered out from the 3,000 genes, 1,550 genes, and we use them as an input to BMD Express tree, conducted a BMD modeling, and then we filtered out 215 genes exhibiting concentration response relationship, and then identified the best BMDL model, which is related to the PLECA7 gene, and it was around 5.7 micromolar. Once we had that data, we then got the fold change related to the expression of that gene, PLECA7, which is here. We converted the concentrations to microgram per liter because it is the input that our algorithm accepts. And we had estimated the free concentrations here as being 2.9% of the nominal concentration. So once we had the free concentrations, which is in this column here, we had our input to the quantitative in vitro to in vivo extrapolation algorithm for reverse dosimetry, and we have done so. And afterwards, we obtained this output here. And in this output, It is important also to mention that the data we have here in this table is a mean of 3,000 individuals. And we have used as well the exposure concentration, which is in this column, the body weight, and the total drinking water to obtain the intake dose in nanograms per kilogram body weight per day. And this allowed us to compare our results with the values that were previously published by EFSA. So after obtaining that output, then we were able to derive a health-based guidance value for perfluorooctanoic acid using the transcriptomics data as input to our algorithm. And we derived this value here, 10 nanograms per kilogram body weight per day. And as long as the PBPK modeling accounts for the inter-individual variability pharmacokinetics, we have used an uncertainty factor of 3.16, which accounts for the toxicodynamic component. And we finally derived this value here, 3.16 nanograms per kilogram body weight per day. And in comparison to previously published values by EFSA related to altered serum cholesterol and to altered antibody titer, they are in the same range. For altered serum cholesterol, EFSA published 0.86 nanograms per kilogram body weight per day, and updated this value for a sum of 4 PFAS, PFOA, PFOS, PFNA, and PFHXS. And this value is 1.14 nanograms per kilogram body weight per day. So on the other hand, we have performed a functional classification using BMD Express as well, just to visualize the most sensitive genes or cluster of genes and the biochemical pathways they are related with. As an example of these, I'd like to emphasize this third cluster of genes here in this plot. And this third cluster is composed by genes that had a BMD median around 11 micromolar. And once we start to analyze the pathways they are related with, we figured out that there are many of them related to the observed health effects caused by the exposure to perfluorooctanoic acid. So as conclusions and next steps, I'd like to emphasize that we have integrated transcriptomics data into our quantitative in vitro to in vivo workflow. We have derived a health-based guidance value in the same range of previously published values. We have also created an in silico workflow that will accept metabolomics data inputs in the near future. We are not proposing here a new health-based guidance value for perfluorooctanoic acid, but only demonstrating a concept and the utility of our workflow. And as a next step for this work, we will try to embed the algorithm in a user-friendly tool such as RVs, which also was developed by Dr. Gerhard in his group at Health and Safety Executive Science and Research Center. So this is the team, supervisory team: Professor Mark Wyand, Professor John Colburn, Professor Ralph Weber from the University of Birmingham, Dr. George Loizou from HSC Science and Research Center, and Dr. Olivia Osborne and Ms. Claire Potter from the Food Standards Agency. Here's my email as well. I'll be more than happy for you to reach out and discuss further this work. And I'd like to thank you for your attention and your time. Thank the organizers of this enjoyable event. I'd like also to thank the Food Standards Agency for the fellowship in computational toxicology, the University of Birmingham and my supervisors for their support. And again, thank you for your attention and your time. I will be more than happy to address as many questions as I can. Thank you.
Great, thanks, thanks, Arthur. And it's really, you notice that the fields work differently, right? We had agrochemicals, pharma comes later. Yeah, here in pharma, right? You can do target-based profiling, for example, right? But that wouldn't really make sense here, right? So I think you can only do a gene expression, for example, approach, right? It really works differently. Depends on chemistry and the field you operate in. Yeah, no, very nice. Yeah.
Yep. Thank you.
Okay. Any questions for Arthur? Okay, let's see. Feel free to raise your arm or let's see. Yeah, any questions from anyone? Let's see, there was one question for paper, but George answered that already, so thanks for doing that, George. Okay, yeah, there don't seem to be any questions, so great. Thanks a lot, Arthur.
Thank you very much.
Cheers, everyone. We will then move to a slightly different area. So that was PK, right? Pharmacokinetics. That was at least part of the previous presentations. And now we move to another acronym, I guess. So MIEs, molecular initiating events. And the question whether we can use actually structure-based methods in order to anticipate and understand MIEs. And Gerhard Ecker will present on that topic.
Yeah, I'm sorry, I'm just...
No problem, no problem. We're actually even a few minutes early. That hardly ever happens at conferences, but here it does. No,
Yeah.
but we can see you and hear you. So from that side, everything is fine.
Okay, yeah, I just need to... there was a recent update of, obviously, and...
Yeah, finishing your slides, yeah? So very good.
No, not finishing the slides, but just...
Yeah.
Zoom doesn't, my Apple doesn't allow me to share.
Okay, because it was, let's see.
Yeah.
In advanced options, let's see who can share all. That should work.
I guess I get the message. I need to just beenden and erneut öffnen. So I just, I guess, step out and come in again.
Yeah. See you in a second.
Yeah, sorry for that. Yeah.
No problem.
So at least you see me. So as you said, yeah, okay.
And the audio is good as well. So everything technically.
Audience, okay. So yeah, getting better now. Just need to again open the file.
So this is fine. I will use those 30 seconds. There was one paper because we discussed the impact of compounds, right, on health. And there was one paper that is maybe a very important one, but the impact of coffee on cardiovascular disease, right? Also, dose is important, so you should have 2 to 3 cups a day. Which coffee doesn't matter so much, can be decaffeinated, ground, and so on. But if you want to avoid arrhythmia, you should have 4 to 5 cups. Yeah? Just posted that in the chat as well, so make sure to have your coffee. So just have in mind as well. Okay, very good. Excellent.
So you should see my slides now.
Yep, all good.
Yes. Okay, so yeah, as Andreas already pointed out, that will be a bit different. We are going to the, let's say, molecular basis, so structure-based prediction of molecular initiating events. And this is the slide I'm basically always showing. More or less, it's we have 2 basic ways how we can somehow come from a chemical structure to an effect on a living organism. One is what I call the, let's say, knowledge-based way. So you have a compound, you try to assess how it interacts with a target. You then embed this target in a pathway, and then you infer from the reaction on the pathway on the living organism. And the other possibility is more or less the machine learning-based approach. You have a set of actives and inactives, and you directly try to build a model and somehow then get more or less predictivity with respect to, yes, an effect or toxicity. So I would like now to focus on the chemical structure and the interaction with a given protein, what we call, and I guess you know that, the molecular initiating event. So when we try to assess a complex adverse outcome like steatosis, like DILI, like developmental neurotoxicity, we build up the so-called AOPs, which start with a molecular initiating event, which means actually a certain compound is interacting with a protein and then triggering an effect. And of course, in a standard AOP, you have more than just one molecular initiating event, and you have key events, and then it goes further till it ends up at the adverse outcome. And the talk today, I would like to focus on, can we do that molecular initiating event, this assessment, can we do that on a structure-based way? That means using protein structures, using 3D protein structures to get a feeling, to get an assessment, how finally, if there would be an interaction or if there would not be an interaction. So what we need for this is protein structures. And I guess you are aware of AlphaFold. So that was a big hype. I posted here the Nature post. It will change everything. AI makes gigantic leap in solving protein structures. Yes, I guess what we have now is we have the whole human proteome, and we can download a model for every protein, a 3D model for every protein. What we did here, for example, is for SLC transporters, and we are doing a lot of work on transporters. We have a NIME workflow where you just load in all the uniprot IDs of the proteins you're interested in, like, for example, the 460 SLC transporters, and then as output you get, for so many of them, I have in PDB a structure available. For so many of them, I have reasonable Swiss model available. For so many of them, I have a template, at least 10 similar PDBs, so I can build a decent homology model. Maybe I have less PDBs, but still I have some templates, or there is nothing in PDB, so neither a proper template with a certain sequence similarity, nor something else. This we did before AlphaFold came out. Of course, now you can say for every human protein, we have a 3D structure in AlphaFold, but be aware, we started looking on them. If you would like to really do the docking, you still need to carefully look on the structure, because AlphaFold, of course, as all machine learning models, the structures are only as good as the underlying training set is. So if there's nothing in PDB, still the structures you get proposed by AlphaFold, you need to invest time in bringing them on a level that you can do structure-based design with them. And the second thing when you do structure-based design is then the other side, bringing the small molecule, docking the small molecule into the protein binding site. And here I would like to briefly guide you through how we are doing that regularly in our group. As an example, I take P-glycoprotein, this drug efflux pump, because we did a lot of work on P-glycoprotein. So actually it's expressed in the membrane. It's sensing toxic compounds while they are diffusion through the membrane and ATP driven via conformational change, just pumping them out, and thus keeping up a concentration gradient between inside and outside the cells. In tumor therapy, this leads to drug resistance, multiple drug resistance, because of course, normally we use quite toxic compounds to treat tumors, but P-glycoprotein is also expressed at the blood-brain barrier, where it just protects our brain from compounds we wouldn't like to see in the brain. And when we started working on P-glycoprotein, one of the main, let's say, challenges was it's polyspecific. So you don't have a classical binding site, you have somehow a binding zone, which means 8,000 cubic angstrom. So doing structure-based design here is quite challenging, because when you dock compounds in, you get hundreds of poses, which according to their scoring value would be reliable. So what we then did, or what we more or less developed, or how are we doing that generally when we approach a new target, we, let's say, validate or we extract our process based on ligand-based structure-activity relationship on QSAR model. So what you see here is we did a lot on P-glycoprotein inhibitors, classical QSAR equations, in that case just correlation with lipophilicity, which is obvious because the excess goes via the membrane, so you expect that you have a sort of a correlation between PGP inhibitory affinity and lipophilicity in a certain scaffold. And here you see, of course, when you do some distinct changes, you have an additional hydrogen bond accept donor here in this region, or you have very bulky groups in the close vicinity to the nitrogen. You see related effects. So either you see an increase of a factor of 10, so regardless what you're doing here on the structure, when you have here this hydroxyphenylpiperidine, you gain an order of 10 in relation to the lipophilicity of the compound. And we use this sort of information to finally select the data set of compounds, which we then would like, I'm sorry, I just need to make that a bit smaller here. Yeah, much better. To select a set of compounds, for example, with a clear SAR. So, for example, we know when we take the basic scaffold, we have this hydroxyphenylpiperidine in, we have the compound without the OH, because we know from our SAR the OH here does not matter, whereas the OH here matters. We have these aryl piperazine derivatives where they are on this correlation line. So it's simply lipophilicity driving it. And we use a set of 2, 3, 4 compounds with a distinct scaffold. So the basic underlying assumption is same scaffold, same binding mode. Could be wrong, but that's the basic assumption we always have in medicine and chemistry. And dock this small set individually, so every compound on itself, placing it in the binding site, and then do a common scaffold clustering. So we cluster the poses according to the common scaffold. So in that case, that would be the structure here. And then we analyze the clusters. If they are able to explain the SAR, we see. So when we did that, when you do that for P-glycoprotein, for example, what you see here is this is the huge binding area. You see some of the clusters we get. And when you finally take each cluster and analyze the cluster, you come up with this basic binding hypothesis, which is able to explain the SAR. So we see this hydrogen bond to the tyrosine 310 for the hydroxyphenylpiperidine side chain, which definitely brings one order. We don't see any interaction with the propanol hydroxy group, which is also according to what we see in SAR. And we have this hydrogen bond from the tyrosine 307 to this carbonyl group. And we know from our studies, the carbonyl group is also important. And we know from the other side, from mutagenesis studies, that these two tyrosines play a role in ligand interaction and ligand recognition. So this is a sort of a binding pose, more or less derived from ligand-based SAR knowledge we gain in ligand-based studies. How do we validate a binding hypothesis like that? Normally, we convert this binding hypothesis to a structure-based pharmacophore model, and then we screen a library, we buy the top-ranked compounds, and we test them and see if we are really predictive. Another possibility we increasingly are using is, can we use the docking protocol for structure-based classification? So here in case of PGP, you see it worked. We extract the data set out of Campbell for 2,000, 3,000 PGP inhibitors, and then you dock them with exactly the protocol which finally leads to this pose, to this validated pose. And then here, when you have the non-inhibitors, the scoring values of the non-inhibitors and the scoring values of the inhibitors, you can take this intersection point and use that as a threshold for just classifying active, inactive. And with this model, we got the specificity of 0.76, which is not bad considering that you are docking in a protein like P-glycoprotein, which has not really a distinct binding site. And having this now in mind, I would like to present 3 case studies we applied let's say, this way to do more structure-based MIE assessment prediction. One is already published, that's mitochondrial toxicity, so that's coming out of the EU projects, tox risk and then later on risk hunter. So there, the question was, okay, we know that rotenone is binding to complex 1. Rotenone is a pesticide linked to Parkinson. We know when you look on the AOP, you see complex 1 inhibition is the molecular initiating event. 4 is one of the potential molecular initiating events for Parkinson. So we had a look, can we make a structure-based hypothesis how rotenone is interacting with respiratory complex 1. Here you see the respiratory complex 1. It's not an easy protein you would like to handle when doing docking. So that's the area where the binding site is. There is no X-ray structure, so you need to do a homology model, so aligning it to Rhodopacta. And in parallel, we were assessing decoylene because you see it's a very close analogon. And the question is also, are they binding in the same way? And might we also expect from decoylene to finally be linked to Parkinson? So using this common scaffold clustering, that means we took rotenone and decoylene into complex 1. We analyzed the different clusters. In parallel, we also did the ligand-protein interaction fingerprints, calculated the RMSD differences between the different clusters, and finally ended up with a binding hypothesis, which looks reasonable. So you have hydrogen bonds. There's a nice overlay of the 2 basic structures. Question is, how can you validate that? First, we did a structure-based pharmacophore model. So out of this complex, we extract a pharmacophore model. When using now this pharmacophore model, so you see mainly hydrophobic areas, features, and hydrogen bond acceptors. When using this pharmacophore model, we are doing all the pharmacophore models with Ligand Scout and screen 3 different databases. So normally when we do that, we screen drug bank. Maybe if we get some hits, might be hidden toxicity in these compounds if they are suddenly popping up. So there was not a single hit in drug bank, but there was only also rotenone and decoylene as a hit when we screened the data set of compounds we extracted out of literature showing mitochondrial toxicity. So that's a larger data set, and we just had rotenone and decoylene as hits, which finally tells us the pharmacophore model is too specific. Also, when you screen ChemSpace, which is a very large library, you only get 470 hits, which is not what you would expect when you do a pharmacophore-based screening. So finally, what we did when you compare now, when the pharmacophore model is too specific, you make it a bit less specific in the way that you delete some of the features. So when you compare it with this one, we actually deleted these 3 hydrogen bond acceptors to make the model less specific for rotenone and decoylene, because otherwise I just could do a chemical similarity screen for rotenone. And in parallel, we build up a random forest, a classification machine learning model using this mitochondrial toxicity dataset, mitochondrial toxic, non-toxic, and merging, let's say, getting a hit list from the pharmacophore modeling, and in addition then merging this hit list with predictions from the machine learning model and combining a consensus list of compounds we bought, and with our collaboration partners, Bob van der Waart and Marcel Leist, a small set of compounds were tested, and I guess 3 out of 10 or 15 were active. So this shows us that this combination of a structure-based pharmacophore model, which is a bit more relaxed, and machine learning is predictive in a way to predict, at least with certain accuracy, mitochondrial toxicity, or to be concrete, binding to respiratory complex 1. So this you can read up in the papers of paper Florentina Troga. The next use case I would like to show is related to developmental neurotoxicity. We started that also in EU tox risk, and that has been done by Karen Grillberger in my group. So there the question was, it's related to nicotinoids, neonicotinoids. So when you take one of the AOPs for developmental neurotoxicity, there are several out. One is focusing on the nicotinic acetylcholine receptor. So the molecular initiating event is binding to the nicotinic acetylcholine receptor, and then the key events going further, altered cellular phenotype, and at the end of the day, you end up with delayed mental development, schizophrenia, epilepsy as toxicity endpoint. So when talking to the nicotinic acetylcholine receptor, that's also quite a challenge. You see here, it's a pentameric. It's like the GABA receptor. We did some work on GABA 10 years ago. So it was a bit familiar to us. You have these 5 subunits, could be composed of different, so alpha beta, or just so heteropentameric. Alpha 4 beta 2 is one main heteropentameric receptor in the brain, or you have homopentameric alpha 7. And what does this mean? Actually, you have subdomain binding interfaces. So finally, what you're doing is you are talking into protein-protein interfaces here. And of course, you have here, when it's alpha 4 beta 2, you have always a plus and a minus interface. And you have also several possibilities. So you take one of these potential sites and then try to dock your compounds. In that case, we focused on nicotinoids and neonicotinoids, insecticides. You know, these also compounds which are always discussed also in media about being toxic to bees, but also having developmental neurotoxicity in humans. So it's nicotine and this nitroimidacloprid, for example. On the side of the nicotinoids and neonicotinoids, you have imidacloprid, dinetofuran, and a set of compounds. So we have around a set of 15 to 16 analogs from this series with activity data, biological activity data on respective subtypes. So you can start with docking them into the respective binding site. What you see here is what is helpful. You have always this chloropyridine, which we finally use as an anchor when doing the common scaffold clustering. Although also here you see most of them have a quite decent common scaffold, which you can use when you just dock into the binding site to find clusters where all the compounds have more or less the same alignment. And what we came up with, a hypothesis, is when you compare imidacloprid and so that's the yellow here, and this nitroimidacloprid, here's the chloropyridine. So you see it's well aligned. And interestingly, what we saw is this switch, this 180-degree turn of the imidazoline. You see here, it's when the nitro group is there, it's pointing towards this area, and when it's the des-nitro, it's pointing towards the other area. And when you do the scoring, and you see that in structures which are in the PDB, and you get the same when you dock to the human nicotinic acetylcholine receptor. So you can somehow, via docking, reproduce basically what you see in crystal structures of insecticide, of insect acetylcholine binding proteins. And then it comes to, and this is also something I would like to outline, scoring. So the question is, can we reproduce somehow the activity differences, which are almost a factor of 1,000. And here, just to show you what we saw, it's depending on the scoring function you're using. So when you use the glide docking score, you get a very well represented ranking of the activity values. So this nitroimidacloprid being much more active than the imidacloprid. When you use the IFD score, it's less pronounced. Also, when you use the prime energy, it's less pronounced. So we are also playing a lot around with which scoring function can we use when we have a certain problem. So we tailor also the scoring function to the problem, which finally requires data. We have here one additional problem, and that's that there is literature that when you do photolabeling experiments, you get a sort of an inverted binding mode. You see that here it's the chloropyridine showing down and the imidazoline with the nitro group showing up. And we could reproduce both potential binding modes when doing docking. The question now is really what is more or less the real one, or maybe we have to face the situation that at the end of the day, there might be two different binding modes for the same compound in the same receptor. Here you see that's work in progress, so that's not published yet. What Karin currently is doing, she is utilizing the data we have for these 17 nicotinoids and neonicotinoids and validating the docking protocol and the structure into which we dock by doing Spearman rank correlation coefficients between docking scores and biological activity values, because we have structures with a water in the binding site, we have structures without water in the binding site, we have structures which are more open to the outside. And here you see when you do this correlation, so when you have the luxury that you have 15, 16 compounds from the same series with really staggered activity, you can also validate your protocol and the protein model you are talking to via this ranking correlation. I never would do a real correlation between a scoring value and PIC50, because those who are doing structure-based design know that this normally not really works. But when you do a ranking scale, so it is able to reproduce the ranking, and here you see basically there are clear, there's a clear hint that docking into this model and docking into this model finally is best able to reproduce the ranking of real experimental values. And the third approach, which is just started by Claudia, is we take an AOP for steatosis. Here you see a steatosis AOP. So these are the molecular initiating events. You see a lot of nuclear receptors, so also not easy to dock, and combine different methods, combine machine learning based on a data set and individual QSAF models for the individual proteins in the molecular initiating event level. So making ligand-based QSAF models for every nuclear receptor and combining it with a steatosis plus minus machine learning model. As a second layer, doing the more systems biology approach, so creating compound pathway interaction fingerprints. I can send the paper of Barbara, Drug for Path, where we extract from Campbell for every compound the interacting targets, for every target the next shell of interactors, and then look for enrichments in pathways. And so you can create for every compound a compound pathway interaction fingerprint, and you can use this fingerprint for doing machine learning. And the third level will be, and this is Claudia currently working on, to do docking-based fingerprinting for every of these nuclear receptors. So we have 20 proteins, and we finally have experimental data for a set of compounds. For some of them, we try via docking, via reverse docking, to fill this data matrix, also as a sort of a fingerprint, active, inactive, and combine this with the other two methods and see if finally we get more predictive when going for steatosis. The challenge here is Claudia just started, and for example, for some of the nuclear receptors like PPR gamma, you have more than 200 structures in PDB, and you need to start checking and assessing which one finally is the suitable one, or you need to do a sort of a consensus docking into, or sample docking into all of them. So this is what Claudia is currently looking at. So just to sum that up, when we go for structure-based docking, because the question where are we, I guess what on my feeling we need is enough experimental data to be really able to validate the docking pose, and not just using scoring values. So when we have this, at least in the mitochondrial toxicity and neonicotinoid setting, we know it works. What we also need, and that refers to AlphaFold, when we have a selected AOP, let's say with 20 structures, we need enough structures in PDB, on my feeling that we can go forward. Just taking an AlphaFold structure and using it for docking, I would not recommend at that level, but maybe with future AlphaFold versions, that might change. So I hope I could give you a short overview what we are currently doing in this area. And I would like to thank my group because they are doing the work. Karin and Claudia is already they are already on the picture. And this I showed you because that's an older group picture. Here's Florentina. She did the microtox, and here's the funding. And thank you for listening.
Yeah, thank you, Gerhard. Much appreciated. So where do you see, because you talked about pharmacophores, right, docking-based approaches. I mean, on the scale from rationalizing what you see, right, to predictive methods. But you answered that question on your last slide already. Yeah? You need a proper structure, and AlphaFold with sidechains sometimes isn't that good, and so on, right? Proper structure, and of course, docking is more resource intensive, right? If you do it across the chemical space, do you see it more as rationalization or as a predictive method with sufficient precision, recall accuracy or so to be predictive as well? Where would you locate the methods you discussed?
I would say in rationalization. So for example, what I showed with the structure-based classification, it worked for PGP. It did, for whatever reason, not work for BSEP. So we tried the same for BSEP. It's also an ABC transporter, ABCP11, so same architecture. We had a lot of experience, had a high quality data set from AstraZeneca, simply didn't work. So I would, if we get predictivity, I then would like to go back and see if we can now rationalize on a larger scale, because in Risk Hunter, we get a lot of requests. And if we continue like that, let's say one PhD for one protein and one compound class, it will take us ages.
Yeah, that's right. I mean, it's because I'm more familiar with docking in the context of finding bioactive compounds, yeah. And there, one of the things is poses, all right, might be suitable, but scoring is difficult, yeah. And if you then have models you do not trust and white chemical space, so not analogs, yeah, and then the whole thing gets quite shaky in some cases.
Yeah. It's really a try. Let's see if we can. I never thought it's possible to do a structure-based classification with PGP. So. Let's see where we end up, and I'm very curious on the progress of Claudia, because steatosis is okay. Maybe cholestasis would be easier, but steatosis, we have clear MIEs, and I'm keen on comparing the more structure-based profiling with the compound pathway interaction fingerprints and the classical machine learning model, and really see the comparison with the same data set.
Yeah, that makes sense. Okay, any questions or comments from anyone else? Let's see, there's nothing in the chat, but let's see if someone raises his hand. Yep, don't see anything yet. Let's see. Okay, yeah, I think, okay, very good. No, excellent. There's no further questions then. Thanks a lot, Gerhard, and thanks a lot to all the presenters in this session. So we'll continue in 2:30 UK time, so in 17 minutes. See you in a bit then, with applications then from different types of industries. See you in a bit. Thanks a lot.
Thank you.